All right. All right, while everybody's joining, just a few notes on how things are going to run today. So we have about an hour to present an update on our RPGRIP1 program. We are recording now, just an FYI to everybody on the phone. The line is recorded, the video feed's recorded, and the intent is that we will share that on YouTube. Odylia does have a YouTube channel that we're starting to populate. So if you miss the meeting, or if you want to share it for future reference, we will have that up probably in about a week or less, but we'll also post on social media when that's been uploaded. If you have questions, everybody is on mute except for myself as the host, and I have two co-hosts from Odylia today. That is Heather and Catherine. They are on the line as well, and they may pop up later so you can meet them. If you have questions, please submit that through the chat function at the either top or bottom of your screen, depending where that shows up. And then we will hopefully have time at the end of the presentation to open up for, we'll go through what's been posted in the chat channel. And then if we have time, depending on the number of participants as well, we'll allow people to ask questions live that we can answer. If we don't have time, or we run out of time to get to your questions, or you think of something later, there's two ways to get a hold of us in the future. So we do have a way to reach out over email, which I'm going to post that email at the end of the presentation, but it is info@odylia.org. We are also hoping to schedule a town hall style presentation in the future so we can have more of a back and forth conversation and answer your questions. So please feel free to reach out in the meantime between now and then when we schedule that follow-up with any questions, and we'll try to address that in upcoming presentations. All right, I think that's all logistically that I needed to cover. So with that, I'm going to go ahead and start the presentation today. We'll aim for about 40 minutes. I'll try to keep an eye on time, but we might run a little bit over. It's not a hard stop, but we'll try to aim to be done at around 3. And I wanted to start with this initial presentation, if I get everything to respond well, with an introduction to Odylia Therapeutics. I'm hoping some of you have heard of us before, but we are a relatively newer entity, both by when we were founded and started as a company, but also in the style or business model we're pursuing. So I'll go through a little bit more of a background on Odylia, how we're different, what steps we're taking to pursue RPGRIP1 as well as other gene therapy programs that we have in the works. We'll, of course, give an overview of the RPGRIP1 Gene Therapy program, where we are today, where we've been, where we're hoping to go. And that will lead us into timelines and next steps. And then really importantly, how you can get involved. And that can be you as an individual, can be you as a family member, it can be you collectively with the community. And we're also really trying to, importantly, from a grassroots standpoint, expand the network of patients and families that know about the program and also want to help us fundraise for the program. So with that, background on Odylia. So our mission as an independent nonprofit biotech is really how we self-recognize. Our mission is to utilize a unique nonprofit business model to accelerate the development of gene therapies for people with rare disease. This is going to change, in our view, the way treatments are brought from the lab to the clinic. Our vision and our focus currently, we have 2 gene therapies in the pipeline currently in development that'll go into a little bit more detail in one of the future slides. We right now have a focus on really diving into gene therapies in the ocular space. And this really comes a lot from our founding story and the expertise of the team. But the Odylia staff, the Odylia board really brings together the vision of how we can change the paradigm for rare disease. We do have a lot of depth and experience in rare disease, gene therapy, biotech, and pharma approaches to that business model, but really importantly, how we're going to change that and adapt that into a nonprofit environment. So I mentioned we're a nonprofit biotech. We were really founded by 2 primary organizations, and this is important for historical context. Some of you may know about Mass Eye and Ear. We have two well-known figures in the field. Luc Vandenberg is one of our co-founders, and Eric Pierce. Both are on our board and highly active in this program. And then that jointly came together with the work through Usher 2020 Foundation, which does have a focus on developing therapeutics for Usher syndrome, and speaks to one of the other programs we have in our pipeline. So primarily our funding does come from philanthropy as well as biopharma partnerships. And at the moment we're headquartered in Atlanta, but we do have a lot of participation from our board that really spans kind of an international and national and international scope. So while we're based in Atlanta, we work with collaborators all over the globe. We don't have really a central focus in any one region. We consider ourselves an international organization. And to speak to our leadership team, I mentioned Luc Vandenberg as one of our co-founders. Many of you may know his work in the gene therapy and ocular space primarily. And Scott Dorfman comes from a business background, and importantly, he's a father of two children with a rare disease. So I think through this combination between Luc's expertise and experience, complemented by Scott's expertise and experience in both the business world and the rare disease world, came the innovative approach to really kind of rethinking how we develop therapeutics in the rare disease space, given the traditional obstacles kind of dependent upon commercial models. So how do we start to tackle that? Before I go into that, I'll briefly introduce myself. So my name is Ashley Winslow, Chief Scientific Officer for Odylia. I do have a PhD in medical genetics, and I spent quite a bit of time between a few different areas, academia. I've spent some time in industry and really spent probably the past 10 years in the rare disease and gene therapy space. We have a very active, very involved, very experienced, deep set of expertise in our board members and partners. So please have a look. As I mentioned before, Eric Pierce is active in our programs, and I know many of you have seen Eric through his work with the LCA community. So. All right, so how, as a nonprofit biotech, does Odylia really start to change the paradigm of how we develop drugs for rare disease? So this really rests on our understanding and recognition that treatments for rare diseases can be found. The science does exist. And it's the same science that any common disorder or really any other biotech and pharma are looking to capitalize for other more common, less rare disorders. The science is in principle the same. We really focus on breaking down the barriers that exist in the commercial model through innovative approaches to our partnerships. And we start fundamentally with identifying very strong programs where we understand the genetic cause of the disease, where we understand that the science has to some extent been de-risked in the early phases. What we call a proof of concept, usually you have a genetic model of the disease, like a mouse model, and the gene therapy, or in principle, very close to that therapeutic, has been applied and shown to be effective in that model. So we don't start at the earliest phases usually. We really like to start with those promising programs that have a launch point for really going quickly and working off of the proven science. So once we have identified those programs, we build a strategic scientific and business path forward for those programs. And we, again, start to focus on what are our obstacles to moving that program forward. And that's through looking at partnerships with patient groups, with pharma and industry, really trying to have conversations early with the regulatory environment. So in that case, this case would be the FDA or EMA in Europe, for example. We work with academic labs and academic centers, and we have collaborations both in the US as well as overseas. And as well as trying to work with service providers and vendors to really leverage our nonprofit status to reduce costs. So in those partnerships and exploring those partnerships, each program can look a little bit different in how they're executed. And I think I covered much of this, much of what we're trying to focus on and trying to recreate how we develop therapeutics for rare disease is a different blend of science and innovative business models from a much earlier stage, and really trying to de-risk programs at the key stages that they usually get stuck at. So some of those key stages, for example, are when you move from an academic lab that has really promising science, we get stuck in what's called the valley of death. And that's often this late stage preclinical point that is critical for moving programs forward and gaining pharma or biotech interest in that program. So that is right now where we are focused on our RPGRIP1 program, and where we're focusing our efforts to really break down those obstacles and barriers. And just a glimpse at our current portfolio. So we have 2 therapeutics currently disclosed in our gene therapy pipeline. That is our RPGRIP1 treatment for LCA6, and then our USH1C gene therapy to treat vision loss and Usher syndrome. And then I'm not going to talk about it in detail today, but we do have another side of Odilia's focus is on bridge solutions and working with patient groups at earlier stages of development, depending on the research goals of that organization. We have more information on our website if you'd like to learn more. All right, so why I think most of you are here, that was a bit about Odylia. To go into more detail about our RPGRIP1 program. And I'm gonna pause real quick and ask that Heather or Catherine please post the email sign up link in the chat. So if everybody has a chance to look at the chat, if you want to learn more about Odylia, if you want to stay up to date on our programs, please feel free to join our email list. Okay, so I think that many of the families and patients that have joined us today know a lot about the clinical aspects of LCA6. That being said, there is an association of RPGRIP1 mutations also with CORD13, also that's cone rod dystrophy 13, and diagnoses of juvenile retinitis pigmentosa. So it's something that we're mindful of, that we're not just talking about one singular group, but we are talking about a few different diagnoses that are affected by RPGRIP1 mutations and therefore would be treatable with this gene therapy. Now, even within the LCA6 diagnostic category, I think if you look at the table in front of you, these are the primary symptoms that when you go through the literature, you look at publications, these are often the characteristic descriptors of LCA6 that are listed. And usually with an early age at onset, early in infancy or before the age of 1 even, but that age set's recognized to be very variable. So this is a really nice snapshot when we think very clinically of singularly what is LCA6. But in practice, when we talk to patients and families, the key clinical symptoms, the age at onset, differences between siblings in the same family that might have LCA6, these characteristics can be incredibly variable. And this is really important for our understanding when we develop gene therapies. Having a clear understanding of the variability in age at onset, in the progression of the disease, in the key symptoms that define that early onset versus maybe later years of progression, having a clear picture of that variability helps us to better plan our clinical trials. It helps us to focus on key endpoints that are better paired to the patient population at the key stages they are at. So I put this up there not because it's foreign to anyone. Probably, again, the patients and families on the phone know this well, have seen these things, recognize them, but it's showing a snapshot of LCA6 in a very simplistic form. And we recognize that having a better understanding of the variability really helps things. And so that's really a plug for participating in research in natural history studies wherever possible, because it allows us to broaden outside of just this clean, simple snapshot, which isn't always the big picture or the complete picture. So starting to dive into our details on our gene therapy. So the focus for our gene therapy is on a route of administration that would be subretinal. This is the same procedure that is done for Luxturna. It has been de-risked surgically from that standpoint. Fairly common procedure. We don't see any risk with a subretinal delivery, and it really allows us to target photoreceptors, which are key and important for LCA6. The other question we often get is about intravitreal injection, because this is seen as a little bit more appealing to patients than a subretinal delivery. But again, our focus on subretinal for this program is really the opportunity to reduce inflammatory response. That subretinal delivery of the gene therapy does seem to be far more effective in increased expression of RPGRIP1, as well as really targeting the cells that are most important for this disease, and reducing the inflammatory response that you see compared to intravitreal, which really doesn't hold those same characteristics, at least from the data we've seen to date. Many of you might know this, but we are speaking to RPGRIP1, which localizes to the cilia, and again, targeting really photoreceptors. So the ciliar structure is really important and vital for photoreceptor proper function and visual function. What you will see here is retinal anatomy. Here's our outer nuclear layer, inner nuclear layer. And then if you go across, we're looking at this mapping onto a cartoon to try to simplify that structure, make it more apparent. And here we have the connecting cilia structure, and this is where we find RPGRIP1. And if you go across, and this is actually a photo from the mouse retina, we're looking at the actual, this is staining of RPGRIP1 within a mouse retina in red here. Now, our program rests on the delivery. So we have our subretinal delivery injection to that subretinal layer in the eye of our AAV gene therapy. So there are many, many types of AAV out there, and you'll hear words like vector or capsid used pretty interchangeably with AAV. AAV is the umbrella group for all of these different capsids, but there's different types. So our program utilizes the ANK80 capsid, which was developed in Luc Vandenberg's lab. Some other capsids you may have heard of would be AAV2, which is used for Luxturna. AAV8 is pretty commonly used in ocular programs. Why we're using ANK80 for our RPGRIP1 program is we do see increased expression in photoreceptors compared to other capsids. So for the data that we've looked at to date, we see increased expression and delivery to the photoreceptors compared to AAV2, 8, 9, and 5. And to give you an idea of what that looks like, these are pictures we have pictures from both mouse and non-human primates. So there has been pretty extensive testing done and published on ANK80. What you'll see in green is delivery of a green fluorescent protein by the capsid. So in this case, instead of delivering RPGRIP1, we're delivering a green fluorescent protein. So this study looked at the delivery, the effectiveness of delivery to the eye of that green fluorescent protein by different capsids. So we see less with the AAV2, medium amount with AAV8, and really a lot more with ANK80. This is a cross-section, this middle picture here, cross-section of the photoreceptor layers, just showing the same thing. You really have a lower expression level from AAV2, and that increases across 8 and ANK80. And this is just another visualization of different cells within the eye showing increased expression. So we really do feel for a number of different reasons. Also, I think some of them we're still gathering data on and can't speak to much detail today, but we think there might be a reduced inflammatory response with ANK80 compared to other capsid, which is really important for safety and being able to lower doses within the population. Okay, so here's the meat of our presentation, what I really wanted to share with you guys today. We're really excited about the progress the program has made over the past few years. So I'm going to go ahead and summarize that here so you know up front, and then I'll talk about that in a little bit more detail with some pictures, but I didn't want to overcomplicate the real meat of what we're excited about. So our RPGRIP1 gene therapy, we are in preclinical testing right now. So we have not moved yet to clinical testing, which would be our initial human testing. This has been testing done to date in just mouse models of the disease, as well as we have done some preliminary non-human primate testing. So I know we're being very upfront and honest. Sometimes there's sensitivities to animal testing, but to be very transparent, non-human primate testing is monkey testing, and this is a critical part of safety testing before something as invasive and risky as a gene therapy goes into humans. So it's I know that not everybody's comfortable necessarily with that discussion, but when I do say non-human primate, that means monkey testing. So the initial studies in our LCA6 mouse model have shown really exciting, strong results. Actually, that also confirms some of the findings that were done by Luke and Eric as well before the program moved to Odylia. So with the delivery of our ANK80 delivery of RPGRIP1 into the eye, and this is mirrored through subretinal delivery, same as what we would do in a clinical trial, we do see that the RPGRIP1 protein is detectable and that it's localized to the appropriate part of the photoreceptor and retinal structures. So in those pictures I showed before, it's occurring in the right place in the eye where we would expect it. There's also a clear dose-dependent increase in expression of RPGRIP1. So we know that as we increase the amount of RPGRIP1 we're delivering via increased doses of our ANK80 RPGRIP1, we see a very clear increase in that protein. That means that the increases are specific and not due to other processes that we don't understand. So this is, it's probably relatively, it maybe doesn't sound that important, but for drug development, you really want to understand how increases in your dose affect the expression of your protein, and then eventually the effectiveness, which we also see here. So it's not just a dose-dependent increase in the protein, but there's a dose-dependent change in the effectiveness as well, which is lovely. So effectiveness of the RPGRIP1 gene therapy, we do see protection against retinal degeneration and preservation of the outer nuclear layer, as well as the inner nuclear layer thickness. And we do see improved ERG. So we're not just looking at does it occur in the right place, but it actually occurs in the right place and it functions as we expect it to. So it actually decreases the manifestation of the disease within these mice. And then, as I mentioned before, we have done some preliminary testing in monkeys. So these are not a disease model. We generally don't often have disease models in monkeys. So these would be normal primates where we do safety testing. The focus is on, can we deliver the gene therapy and detect it? And is it safe? And this is done in monkeys because monkeys are most similar to humans. Mice are too distant to really, really gain as much faith as you'd like in the safety testing. So we did find in a short-term study that the delivery of RPGRIP1 by ANK80 was well tolerated, and there are no clinical findings related to the gene therapy. And we do see, again, really importantly for drug development, a dose-dependent increase in the delivery of the RPGRIP1. And that was measured in a few different ways. We're really excited about that data. So I'm going to go into that in just a little bit of detail to give you an idea of what the data looks like. So I do love visualizations. Happy to go into more detail if this is foreign to anyone. Let me know if anyone wants a bit more description. But again, this is looking in the mouse eye after delivery of RPGRIP1 by ANK80. And we've gone back and fluorescently stained. I know this might be a little bit foreign, but you can basically go in and detect the RPGRIP1 that's been delivered and make it fluoresce red. And that's where you see here, this is occurring at the right area in the eye, in the right structures. There's another protein that's stained at the same time that detects the cilia structure, so that we know that the red should occur at the end of this kind of green structure. So that's what you're seeing here. This is great. This is really exciting because it's going to the right place and it's expressing at a detectable level. And then just a glimpse at some of the data for function. Some of you may be familiar with this from looking at the data. These are the same types of data that are collected on humans to look at the cellular structure in the eye. So we look at the photoreceptor layer thickness by OCT. And if you can see, these are representative images from our mouse LCA6 mouse model. This is a normal mouse, and the red line here denotes the thickness in a normal mouse. Our LCA6 model has lost RPGRIP1. We've knocked that out to simulate the same disease manifestation in humans and patients, and we see a decreased thickness. And then if we put ANK80 delivering RPGRIP1 back in the eye, injected into that subretinal space, we do see a preservation of the thickness compared to how that's thinned here. So hopefully you all can see that. We do have bar graphs, and this has been extensively studied in many, many mice, but I didn't want to overcomplicate the message today, but happy to go into more detail if anyone has questions. And then to look at function, many of you may be familiar with ERGs. So this is looking basically at the electrical response in the eye. We look at two primary readouts, the A wave, which is the measurement you see here, and then the B wave, which is this big change in amplitude here. Our most significant finding really was in the B wave, but we do also see changes in the A wave. That I'm going to try to explain. Sometimes these line graphs get a little bit confusing. In red is our highest dose. So the biggest delivery of RPGRIP1. This should be our highest expression of RPGRIP1 in our mouse model, followed by, lost my mouse for a second, the green line, which is a medium dose, and then the blue line is our lower dose. The open circles here are our vehicle, meaning we didn't deliver any gene therapy. And so this is our basic baseline function, or really dysfunction in the mouse model. So we do see a regain, especially in this dose-dependent manner. So here's the medium, and here's the highest dose, so showing the biggest regain in that B wave measurement. So we're really excited to see this. The models we're working with are incredibly aggressive genetic models. And having worked in the gene therapy space for a long time, you don't always, even with a good gene therapy, you don't always see a functional recovery at the levels we're seeing. There can often be limitations to working with either very small animals or very aggressive models that don't simulate the human disease as precisely as you hope. But these are really solid data. We've worked with some of our advisors and looking and digging into these data, and they're very impressed and excited about the potential for this program. So with that, I would like to talk about timelines and next steps. So where are we with the program and where are we going? So the program was originally with a biotech partner for the past year and came back to Odylia at the beginning of 2021. That was a bit of a surprise for us, honestly. It made a lot of progress during that year and a half it was with that biotech, but it came back to us not because the science looked great, but I think because some of the business model commercial challenges that we normally see with rare disease. So with that in mind, we've put together a full assessment of all of the data collected for the program to date. We have a full development path mapped out and vendors identified, and hopefully we're ready to move forward as quickly as possible. And that's what I want to talk to you guys about today. So where have we been? The completed and green checks at the top, we have mouse expression study data, we have mouse efficacy data, we have non-human primate or this monkey feasibility study, which is a wonderful precursor to showing that this drug is not just effective in the disease model, but looks to be safe. But we need to draw that out into what the FDA or other regulatory bodies would recognize as real extensive safety testing. So that's where we're going next. I'll go into that in a little bit more detail. We have actually started our interactions with the FDA through an orphan drug designation and rare pediatric disease designation application. So we are hoping to get feedback on those applications later this year and early 2022 at the latest. I think the latest deadline we expect is about January 2022. The initiated, I'll go to next, the yellow check marks here are manufacturing, and that is actually manufacturing of the gene therapy itself, which for gene therapy is extremely complicated. Gene therapies are a biologic, which are different from a chemically based manufacturing process that you'd see with like Tylenol or Advil. So these can be big, lengthy manufacturing processes that the FDA likes to see a lot of detail on. And there's a lot of things that can go into affecting what we call the yield within the manufacturing process. So happy to answer questions if people want to dive into more detail on that. But we've started to look at the contracting for that piece of work, as well as started our engagement for clinical development. And I'd say really the focus of my next slide is on our next step activities for our formal toxicity study that we have to do to prove safety. We have a few other regulatory interactions with the FDA along what's called a pre-IND meeting, and then our formal IND submission. So once you do the IND submission for the program, this is our last step, and this would open the conversation with the FDA for clinical trials. These are our projected timelines for most of these activities. So preclinical, again, referring to the focus on animal work, and this would be on assay development that allows us to move into that formal safety toxicology study. And then that would round out our preclinical work. In order to do the toxicology study, we have to have our gene therapy manufactured at a level that is the same process that we would use for the clinical product. And that means that how the gene therapy is manufactured for use in humans, we have to have an almost identical process for that's tried in the safety testing in monkeys. So these, again, it's a biological process. It can be much more complex as far as a system than your usual kind of chemistry-based manufacturing. And these timelines usually stretch in the early toxicology-focused production about 8 to 10 months. And then we move into clinical grade manufacturing, which can stretch for a similar period of time. We have completed some of our regulatory interactions, as I mentioned in the previous slide, but we have coming up a pre-IND meeting that we're planning for. And then ultimately to open the program for clinical trials would be an IND submission, which is our last step. And I'll mention real quick, we're really excited. We have a great clinical advisory group, which I'm going to share with you in our next slide, but we have started work with them. We have reviewed the preclinical data to date. We started thinking about trial design, what our treatment window looks like, critical questions about, some of the variability I was mentioning before and how we take that into account, because you don't always get a clear picture just from the literature. So this is where it's really important for us to talk to clinical advisors as well as patients and families along our clinical development activities. And here's our clinical advisory group that we've started working with. All of these advisors you'll recognize maybe some of them from having worked with the RPGRIP1, either in a preclinical understanding. Tien-Tsin Lee has been one of the earliest researchers within this space and really a huge advocate for understanding the disease in the animal model and how it manifests and how that compares to the human condition. Eric Pierce, many of you know and have met with personally. Ted Dreija also has done a lot of research both within the RPGRIP1 clinical and preclinical space. Same with Rob Konenkoop, who's based in Canada and has seen many patients within the community and currently is very active. And Jiang is at Emory, has a huge amount of depth in the genetics of ophthalmological disorders, as well as very deep experience in surgical procedures and working with different companies on new and innovative delivery mechanisms. So we're really excited that this group has agreed to work with Odylia and is so excited to lend their expertise to the RPGRIP1 program. And then my last slide, I'm going to hand it over to Heather after this. Next steps. So the remaining development costs for our program, those that I outlined in the previous slides, really we estimate at current value to be about 4.3 million to take us up to that IND submission. The key components of that very large number, I'm guessing for many of you that seems big, fairly standard in biotech and pharma terms for where the program is. But the three main components that really go into that number are our ANK80 RPGRIP1 manufacturing, our toxicology and biodistribution study, so that last final safety study, and then assay development to support multiple functions of the program. So our assay development supports manufacturing, toxicity, as well as our clinical studies going forward. We estimate that if we had that money tomorrow, we could move the program forward within 20 to 24 months. There are opportunities, I think, with the right type of partnership or funding to even accelerate those timelines, but trying to be fair to the numbers and our discussions with the different partners and vendors and service providers that we've identified for the program, we're probably looking at about a 2-year horizon once we have funding. And then Odylia is pursuing all routes towards funding and reducing costs for the RPGRIP1 program. As I said, much of what we're doing at this phase is trying to focus on what are our obstacles and how do we remove those and how do we ensure there's a strategic and scientifically sound plan for the program. So we have spent quite a bit of time in the past 9 months once we became aware that this program was coming back to Odylia, focused on identifying potential commercial partners within the biotech and pharma space, or maybe even in venture capital. We've been searching also for innovative kind of hybrid models for co-development with potential partners. We've been applying for grants. We are right now really ramping up activities around philanthropy and fundraising. And that's why we really wanted to connect with the community today, both to give an update for where we are and get questions and try to understand as a community what questions you have about the program. But also to get you involved, because we've had a lot of people reach out and say, we'd like to help and what can I do? And then we're also working to identify service providers and vendors for the manufacturing piece and the toxicology piece that kind of get that as a nonprofit, we can work a little bit differently together. So we're looking for those strategic partnerships. And I think we've identified those. We have plans to move those forward as soon as we have those funding pieces in place. So with that, I'm going to go ahead and introduce our Director of Development, Heather Green, who's going to talk to you guys a little bit more about how you can get involved.
Great. Thank you, Ashley. Can you do the next slide? Well, first, let me say we're very excited to have y'all with us today and to be able to share information about the program for RPGRIP1 and what we are doing to move this to clinical trial. The community that we estimate for our RPGRIP1 is about 400 to 600 people in the United States, and worldwide as many as 20,000. But there are just a fraction of those people, obviously, on the call today. So one of the first things that we need to do is expand this group to let more people know about what we're doing and what the program we hope we'll be able to do, and just to raise awareness and ultimately raise funding. So one thing that you can do is sign up for our newsletter. We put the link in the chat, but while you did register today for this webinar, we did not add you to our email list because we want people to opt in and not automatically get added. So this is a screenshot of our website, and you can go to the website and navigate under our impact and get involved, and you can sign up for the newsletter. We send periodic emails, and then we have an e-newsletter that right now we send out twice a year. So we encourage you to do that, and then to share that with other people who you think might be interested in what we're doing. Encourage other people to via social media. So if you are already following us on our social media, fantastic. We're on Twitter and LinkedIn, but also if you aren't, go ahead and follow us there and then share some of what we're doing. We try to post things there once a week or more. So if we have new developments or other articles or things that are going on, that's the primary place that we share them. And then maybe y'all can help us share that as well through your networks. And last, but I'm sure there are other things that we can do to raise awareness and bring more people in, is let your patients' providers know as well. So if they don't know about the program that we have, let them know. We do have a one-pager about this program, and I'm happy to send that to you. So you can reach out to me and or our info email, and we would be glad to send that on. So that will help us get more people aware of what we're doing, and ultimately, hopefully we can raise the money to move the program ahead. Next slide. So of course, if we had enough money today, we would move ahead in the program. We could be at IND in 24 months. But we are going to take this one step at a time. And so we've sort of divided it into 3 chunks. And our first chunk is about $950,000. So we are doing everything that we can, talking to anybody who we can about raising that money so that we can start the process for the final toxicology studies, which is the next step in the program. So we are, instead of looking at the whole pie, just that small piece first. And we feel like we can do that. And then we'll move ahead to the next, and we'll raise the next chunk of money. Next slide. So you, like me, are probably saying, how are we going to do this? And we have a lot of ideas, and we also hope that y'all might have some ideas. We are continuously seeking donations from any source that we can, individuals, foundations, companies. Particularly, we're looking at large companies who have an interest in ocular, rare diseases, gene therapy. It doesn't just have to be people who are developing other therapies, but even an eyeglass company, for example, or somebody who we can make a good fit with, and those are probably larger donations, $50,000 to $100,000. And one thought is that not all companies, not all pharmaceutical companies are going to be able to take on a program like this, but every single company can benefit from a treatment in the market for patients to prevent vision loss. Everybody can benefit from that, and that's what we're trying to get out into the world and more companies. And takes a village, and we will work together, and some money from several sources will help us get to where we need to be to reach this goal. And that's sort of how we feel like we can do it. So we're telling our story to anybody who we can. And if you have suggestions of companies who might be interested, please let us know. My email is on the screen. It's hgreen, and green has an E on the end, at odylia.org. But any and all suggestions are welcome. So just reach out to me if you think of somebody who might be interested. And it isn't that we need to ask them right away. We need an opportunity to tell them our story. Everyone that I talk to and about Odilia's mission thinks it's a great idea. And if we tell enough people, we'll find the companies and the support for the philanthropy we need to get to our goal. And as Ashley mentioned, we're a nonprofit organization, so we will leverage that any way that we can in order to lower costs, which means less money that we have to raise, or partnerships that we can create. There's a lot of creative ways that we can raise the funds or lower the costs to get this program to clinical trials. Okay, next slide. So we're hoping after today that we will be able to kind of gauge the community's interest in getting involved, how, if any of you can help us, give us suggestions, or if there's things that you would like to do to help us raise awareness, raise funds. Do you know of companies that might be interested, or other things that we could do to reach this goal? So it's a grassroots effort that will, I think, culminate in us being able to get to clinical trials. I really think that we can and we will do this. It will take many of us working together to be able to raise the money, but it's a great idea, and I think we can make a difference. So if anybody, again, my email is on the screen. So if you have some suggestions or even questions of things that we are doing or other companies, please let me know. And I think that's it.
Yeah, just a reminder, we do have the info@odylia.org email if you'd like to reach out with any questions about the program. If you have specific inquiries about how you can help, how you can get involved in fundraising, what resources are available in order to connect and broaden that network that we're reaching out to. I think some of the lines have become unmuted. Then please reach out to Heather at H.
Oh, wait. Oh, sorry.
No, that's okay. Please reach out to Heather or our info. Please, if you would like the latest and greatest on what's going on at Odylia or updates on our programs, there's a few ways to get involved through social media. We are on Twitter, we are on LinkedIn, we're on YouTube. That's a growing, I guess, database of our presentations, where we will be adding this later. So please feel free to share it. With that, we do have a little bit of time, and I want to make sure we have time for questions today. But again, please send questions through to our info box, and we're going to kind of gauge interest in having a follow-up town hall discussion. So with that in mind, please, we'll prioritize questions entered into the chat box. I'm hoping that those folks who are just adding and kind of spamming the chat could please stop that. So in the presentation, you mentioned improvement in ERG and prevention of further degeneration. So does this mean the patient's vision will improve or remain the same but not get worse? Also, how many doses do you envision one time yearly? So I'll answer the last part first real quick. As far as dosing, with gene therapy, at least the state of where we are today, and the sciences around gene therapy, as well as just our science and understanding of this technology in general, is growing constantly. So where we are today, and what I would say has been put forward as an example of Luxturna, is our expectation is this is a one-time dose, and there's a continuous effect because we deliver the gene therapy into the subretinal space, it's held there and continues to express RPGRIP1 over time. It's not like Tylenol or Advil, like always go back to those examples, that's washed out of your body over time. You have to keep taking it. Gene therapy has a very, very long lifetime. There are groups, and this is where the science is always evolving. There are groups looking at redelivery or second dosing of patients much further down the road. This is really looking at, 10 years or going back in and treating a subset of cells that additionally with more. That is not the plan for this gene therapy. And I would say 99% of gene therapies currently in development, we are really looking at a one-time only delivery. And then as far as whether we are hoping just to stabilize the function that's there at the time of dosing, or whether we expect there to be improvement. To be completely honest with you, many times in drug development, we don't know until we start to work in the clinical setting. So at a minimum, given the data we have today in the animal models, we expect to stabilize the function that's present at dosing. That being said, in discussions with clinicians and other researchers, we do believe that given the state of photoreceptors and also given our biological understanding of dysfunction in RPGRIP1, when we have preserved photoreceptors, but a loss of RPGRIP1 function, we think there's a potential for a regain of function, but we really won't have the opportunity to prove that out until we get to testing this in a human population. That all being said, almost with every gene therapy, the earlier that patients are treated, the more protection and the more possibility for regain of function that we think there is. Okay. Do you know at what age is it possibly too late for gene therapy? Also wondering if there is a trend in degeneration in patients with LCA6. So we know, again, kind of from that, we know there's a lot of variability in the onset and manifestation or presentation of LCA6. And some patients may decline much faster. Some patients may decline differently from other patients. But looking at it from a more simplistic view, with early onset, and we do think that there is a very long treatment tail or treatment window. So even with early onset, we think from the data that's been collected clinically that there are preserved photoreceptors into adolescence and early adulthood. And so we do think that the potential for the gene therapy at this point in time, from our understanding, is that as long as there are preserved photoreceptors, there's the potential for regain of function. But again, always, the earlier you get treated, the better potential there is for preservation. So somebody mentioned they have their Amazon Smile set up to donate to Odylia, which is great. We forgot to actually add that to the slides, Heather. I think that's a great idea. Any way you can think to support us and spread that to your network, that's a really easy one that doesn't take much effort. We do have instructions, I think, on LinkedIn and Twitter. But if you have a hard time finding those, reach out and we can send those to you.
And yes, they do. Ahead, they do help. Every little bit does help, and that money does come straight to Odylia.
All right. Anyone else? Any other questions for right now? We have a few people who are just typing random things in here. So right now I'm probably going to keep the lines muted just so we keep this productive. Again, if you have questions, please put them over chat so we can pre-screen them. Yeah. Can we briefly explain the difference between phase 1 and 2 trials? Happy to do so. So normally in drug development, you have once you get into clinical trials, you have phase 1, 2, and usually 3, and sometimes even 4. Phase 1 traditionally focuses on just testing the safety and getting the dose, a safe dose in humans, correct? Phase 2 usually focuses on proving that it's effective, and then, or getting initial data that shows it's effective. And then phase 3 generally proves that it's effective. What's different for rare disease as well as for gene therapy is phase 1 in most other diseases where you don't have as invasive or permanent as of a delivery of like gene therapy, which is this one time only, it doesn't wash out, fairly invasive procedure, although subretinal is very safe for everything that we've looked at to date. It's because of the nature of it, we don't use healthy volunteers, which in other studies where you have a drug that washes out very quickly, or is less permanent or less invasive, you will enlist healthy volunteers to do that safety testing first. So with gene therapy, that testing will be done first in patients with the disease. And so you have the opportunity to do two things within that first trial. One is to absolutely, number one, you have to prove safety, and that's the focused and primary endpoint of a phase 1 or initial study for a gene therapy program. But because you're conducting that study in actual patients with the disease, there's the opportunity to show if it's also effective in that disease. So oftentimes with gene therapy programs, we will run what's called a phase 1/2 trial, where you combine these two goals into one trial, which means they can sometimes move quite a bit faster. And the FDA has been open to that. We've had many trials that proceeded that way. And it's something that we would probably pursue for this program too. My daughter will be 2 in December. I presume she'd be a late phase for trials. Once trials start, at what point would she be eligible? Projected 2 years after trial starts. So we haven't fully delineated the what we call inclusion and exclusion criteria for the program. The inclusion criteria set the guidelines for who's included, exclusion criteria for who's excluded. So that really sets the bounds on who the focus is for each phase of the trial. And normally you'll see sometimes that inclusion exclusion criteria will change between that phase 1/2 to the next step. Oftentimes with gene therapy, just as general trends, again, we haven't set those parameters yet for this program. We're working with our clinical advisory group. Excuse me, sorry, it was just one of the things prompted for live transcription, or closed captioning, so that now is available for the meeting. We haven't set the parameters for this program specifically, but we will be working on that with the clinical advisory group. General trends in gene therapy, because it's a more permanent invasive procedure, usually these are done in older, either adults or older children, before we go into infantile or very, very early pediatric populations. Similar to what you see with the COVID vaccines, they will work through phases of older adults into younger and younger age groups. Because of the nature of the onset of the disease, we are looking at ways to innovatively work with the regulatory environment, the FDA, to move those trials earlier, as early as possible. But we do think the treatment window, while earlier is better for preserving function, we do think that there is a long treatment window that probably extends into early adulthood as well. And I think I've answered everything, but if I have missed something in the chat, please let me know. And again, please reach out if you have any questions at info@odylia.org. And please reach out to Heather if you have any ideas about how to get involved or just questions. And I think that's it for today. Anything else, Heather, that you want to add before we go ahead and sign off?
No, we will be following up once we set the next town hall for people who do have questions. So we will send you an email about that.
And thank you again for joining. We really appreciate the support. We really would love feedback on whether even just the venue today if this worked for getting some critical information to the community, but we're really hoping to have updates regularly. So please let us know if you have ideas, questions, or thoughts. And thank you for joining.